Hi class, let's talk about the digestive system. Like the other body systems that we have covered, as we're going over this system, we're going to first focus on structures, then we'll focus on normal biota, and there's quite a bit of normal biota within our digestive system. And then we'll spend the majority of our time talking about diseases of our digestive system. So, let's get started with the structures. Here's a list of our structures in the GI tract or in our digestive system. When we see this term GI, I want you to think gastrointestinal. Gastro is a prefix that means stomach. Intestinal, well, means intestine, small and large intestine. So when we think GI tract, I want you to think of it as a long tube. Generally speaking, the GI tract is just the stomach to the intestines, whereas the alimentary canal is going to be going from the mouth to the anus, and that's typically the definition if you go look at an anatomy and physiology textbook. But we're going to be a little fast and loose with our definitions here in a microbiology class. So if we look at those main sections, we'll start off with the mouth, then we travel down to the esophagus. From the esophagus, the food will travel into the stomach. From the stomach, we'll go to the small intestine, which will have our duodenum, jejunum, and then ileum. Then we'll go to the large intestine, which consists of the cecum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and then anus. We also have some accessory organs within our digestive system. We have the salivary glands, the liver, the pancreas, and the gallbladder. The accessory organs are notable in that they do not produce or they do not have food traveling through them, but they make secretions that help us process the food. So we have our salivary glands, liver, gallbladder, pancreas. They add digestive fluids. Our digestive tract, since it has such a high bacterial load, is going to have a lot of normal defenses built into it. These normal defenses help us keep the bacteria within our digestive tract where they actually do some fairly good things for us, as opposed to letting that bacteria out where it can cause a lot of problems. We have a layer of mucus throughout our entire GI tract. This layer of mucus is going to be lining the lumen, or the inner cavity of the GI tract, and helps to resist the digestion of our GI tract so that our GI tract does not digest itself, and also makes it more difficult for bacteria to penetrate the lining of our GI tract. We also have a lot of immunoglobulin A found in our GI tract. Immunoglobulin A is the antibody that's associated with mucus, so with lots of mucus we have lots of this antibody. The GI tract also has some muscular walls in it. These muscular walls are going to have a rhythmic contraction called peristalsis that moves food one direction and liquids one direction through our GI tract. This peristaltic action is the reason why we can drink something while standing upside down doing a handstand. Well, most of us can. It's been a while for a lot of us, though. And that movement of constantly flushing in food and fluids one way through our GI tract is going to make it so that we're constantly flushing the bacteria out of our GI tract. Now, the bacteria that we have are adapted for this. They are going to have very rapid generation times, or they're able to reproduce very quickly so they can constantly repopulate despite the flushing motion of our GI tract. We have a lot of lysozyme and lactoferrin within our saliva. These are two enzymes that help reduce bacterial counts. And it's also one of the reasons why the human mouth is much more sterile, or I shouldn't say sterile, has a much lower microorganism count compared to our canine friends. Our stomach acids, or stomach fluids, are very antimicrobial due to the high acidity, and we go from a pH of about 2 in the stomach to a pH of 8 at the beginning of the small intestine at the duodenum. So there's a huge pH fluctuation that can denature many of the proteins within the microbes that we ingest. And then finally, the bile itself that's secreted by the liver and gallbladder is antimicrobial as well. Within our GI tract, we have some gut-associated lymphoid tissue. We talked about this briefly when we were in the chapters on the immune system. This gut-associated lymphoid tissue is a generic term that refers to any tissues of the GI tract that has an immune function. So if you look at the tonsils and adenoids within our mouth, our upper GI tract, our oral cavity, we have some lymphoid tissue there. We're also going to have very small patches of lymphoid tissue within the esophagus. The esophagus is not noted typically for having lymphatic tissue or lymphoid tissue, but it is present. Within the small intestine, we have regions known as Pryor's patches. These regions are able to sample the microbial count and the microbial population that's in our small intestine. The appendix consists almost exclusively of lymphoid tissue, and it's worth emphasizing this is secondary lymphatic tissue. So if the appendix is removed, we're still able to get by okay. But yes, the appendix does serve a function. It is not vestigial. The appendix helps us with our immune response. If we look within this lymphoid tissue, they're going to contain a wide variety of lymphocytes. There'll be some phagocytes, which help us with our nonspecific immunity, and then there's also going to be some T and B cells, which help us with our specific immunity. Our lymphoid tissue, or gut-associated lymphoid tissue, does produce IgA, and also is going to do a wide variety of other functions. Another defense within our digestive tract is the bacteria themselves. They are very commensalistic in nature. These bacteria, the commensalistic bacteria, outcompete the pathogenic bacteria, making it difficult for the pathogenic, or the bad guys, so to speak, to reproduce and raise their populations levels up to the point that they would cause a problem for us. Our commensalistic organisms that are within our digestive tract have a special adaptation that helps them to survive and remain in our digestive tract. That is specifically that they mimic the sugar coatings of the proteins of our intestinal walls so that these bacteria can trick our bodies into thinking that they belong there. This is kind of an insidious trick, I personally think, and it's mimicked by some pathogenic strains of bacteria to also evade detection within our immune system. If we look in the oral cavity or the mouth, there's going to be quite a few species of bacteria known to populate it. Our mouth is unique in that it has both aerobic and anaerobic conditions present. Some of the bacteria present in our mouth are going to be streptococcal, Neisseria, Villanonia, Staphylococci, Staphylococcus, Fusobacterium, Lactobacilli, Corynebacterium, Actinomyces, and Treponema. There's going to be yeast or Candida albicans present in our mouth, and then there's going to be Trichomonas tenuis and Entomoeba gingivalis for the protozoa that are present in our mouth. So we have a good amount of bacteria present in our mouth. The exact composition and breakdown of the microbes that are present in our mouth remain relatively constant over our lifetime and are going to be similar within members of the same family or between people that spend a lot of time kissing and exchanging microbes between their mouths. The bacteria live on our teeth and the soft structures of our mouth is coated with bacteria. If we look elsewhere in our digestive tract, we find, particularly in our oral cavity, that the phenomenon known as dental plaque. This dental plaque is a kind of biofilm that can form in our mouth. These bacteria that are held in the biofilm have very specific recognition molecules that allow for them to communicate with each other. And one particular strain of streptococci that's alpha hemolytic can colonize our mouth very quickly after it's been cleaned. This is the pioneer species. They produce a pellisol. This pellisol is a mucigenous glycoprotein, and this forms the covering on our mouth that streptococci attach to. So if it's been a while since you've brushed your teeth and your teeth just feel kind of grimy, slimy, you have a pellisol, that mucigenous glycoprotein covering, or that coating of protein mucus on your teeth. And then once that establishes itself, other species of bacteria can attach and start to form the biofilm. If we look in the pharynx, or the back of our throat, there's also another wide variety of microorganisms. We covered that in Chapter 19 when we focused on the respiratory tract. If we look in our stomach, generally speaking, is fairly sterile. That's because of its incredibly low pH. You will find a couple extremophiles that can live in our stomach. These extremophiles are going to have the ability to live in highly acidic environments. Back in 2008, as part of the Microbiome Project, we found signatures of over 100 different microorganisms that can live in different species of microorganisms that can live in the stomach. Some of these microorganisms can just be passing through. If you consume a lot of food and a lot of liquid all in one sitting, the pH of the stomach will be temporarily raised as you dilute that stomach acid. So let's say I drink a big glass of spoiled milk, which I probably wouldn't do because it would taste terrible, but I did it. And the spoiled milk has a pH that's higher than 2, and as I drink it, it dilutes the stomach acid, raises the pH, and makes the pH more palatable, or less hospitable, or less dangerous, I should say, for the microorganisms, and those microorganisms could temporarily be moving through the stomach while the pH is temporarily raised to then colonize the small and large intestines. Some of the bacteria that have been found in our stomach include bacilli, Clostridium, Staphylococci, Streptococcus, and Streptococci residents. Another notable one that we'll talk about is Helicobacter pylori. That's more of a pathogenic microorganism. Within our large intestine, there are a lot of microorganisms. We have billions of microbes per gram of contents in our small intestine. And when I say billions, I mean hundreds of billions. 10 to the 11th is 100 billion. These bacteria that are present within our large intestine, where we have our feces or poop being produced, include the bacteriocytes fusiform, Bifidobacterium, Clostridium, Streptococci, Peptostreptococcus, Lactobacilli, Escherichia, and Enterobacter. When you see this term entero, this is a term we'll see a lot in this chapter. This term entero refers to the intestinal tract. Any kind of enterobacter is going to be a bacterium associated with the intestinal tract. We have those Candida or yeast fungi, and then we'll also have some fungi and archaea that have been isolated or that are present within our large intestine. The normal biota of our gut gives us a lot of protective functions. It's also worth mentioning that it helps us with digesting some of the nutrients that we consume, and these bacteria also produce some essential nutrients that we need as well. So these bacteria help us with digestion. They synthesize vitamin K, for example. These normal biota also help our epithelial cell structure to have the proper function. They help to stimulate the proper growth and regeneration of the epithelial cells lining our intestinal tract. And they also help to teach our immune system how to react to microbial antigens. Individuals that have a gut microbiome that's out of whack, generally speaking, are going to be more likely to have an autoimmune disorder. This mix of gut microbiota is very important for us, and it's one of the reasons why fecal transplants are becoming an increasingly popular therapy for individuals that have an out-of-balance gut microbiome. The accessory organs and the internal organs are free of microorganisms that are part of their normal biota. They can have microorganisms isolated from them when there is a disease state in place, but generally speaking, our gut organs, other than the intestinal organs, so think kidney or liver, are going to be free of microbes. And that is all we have for this section. If you have any questions for me, please feel free to post them to the class discussion board or shoot me an email. Happy studies!